So I'm very pleased to welcome Dr. Wisniewski from the University of Pittsburgh today for our seminar. I've had the pleasure of working with him and his group for the past 6 years on a trial that we're about to finish up, which is exciting too. I've learned a lot from their group at Pittsburgh. He is a professor of epidemiology, psychiatry, and clinical and translational science at the University of Pittsburgh. He serves as a vice provost for budget and analytics and is the co-director of the Epidemiology Data Center. During his tenure at the university, Dr. Wisniewski has developed successful research programs focusing on the design and analysis of clinical trials, specifically multi-center clinical trials in a number of medical disciplines, including mental health disorders such as depression and bipolar disorder, pulmonary conditions such as asthma, idiopathic pulmonary fibrosis, and chronic obstructive pulmonary disease and trauma. He's also done some work in ophthalmology, and he is currently one of the principal investigators of the Coordinating Center for Accelerating COVID-19 Therapeutic Interventions and Vaccines, active for the anti-thrombotic effort, and that's what he's going to talk to us today about. Thank you so much for coming, Steve.
Thanks, Janet, and thanks for inviting me, and good morning, everyone. I wanted to, first of all, pass on apologies from my colleague, Frank Sherba, who was supposed to be joining me today, but he got tied up, pulled into something else, and he could not make it. So he did want to apologize for that. What I'm going to give you today is a very broad overview of this very large initiative that NHLBI has put forward. And what I want to try to talk about is not so much some of the specifics of a trial, like working it soup to nuts, but more some of the novel design considerations that we had to think through in trying to do this in the context of a pandemic. So I'm going to try to give you a little bit of an overview and then jump into some of the specifics. My goal is to obviously finish with plenty of time for discussion and questions. So why don't I just jump into it then? So what I first wanted to do is just give you one background. I mean, it's kind of silly at this stage of the pandemic to feel like we need to provide a background slide on COVID. But the academic in me feels like we have to follow a certain format. So I'm just going to make sure that I cover the background stuff. So COVID is a novel coronavirus that came about in 2019, first reported in Wuhan, China, and then as this was coming about right around last year at this time, as we were moving into quarantine. It was become pretty clear that effective vaccines were not developed, and that hasn't been until recent. And so the government, and particularly the NIH, thought it was very imperative that we look at secondary prevention strategies and evaluate these as quickly as possible in order to minimize disease burden. So what we were starting to look at, treatments that target multiple disease exacerbating pathways could present unique opportunities to augment currently clinical available treatments for COVID. And we thought the impact would be worldwide, thinking that, as of May of 2020, there were a huge number. And I'm post-dating this to May because I'm trying to give you the thinking of what was going on as we were initiating this program. So you can see that, and everyone is clearly aware, and this is taken from the Hopkins site from about a week ago, I believe. I can't remember when I exactly scraped it, but you can see that the burden is massive, not only in the US, but around the world, where they're nearly approaching 3 million deaths and well over 124 million cases at this stage. So one of the things that when the NIH approached us about this, and specifically the NHLBI, they wanted to talk to us about bleeding and clotting in COVID-19. So COVID-19 is associated with a thrombolytic state marked by some thrombosis, neutrophilic infiltration in the lungs, some vascular leaking, and development of acute respiratory disease syndrome, or ARDS. A lot of the complications are exceptionally common. We thought at the time it was unclear really because we were at the beginning of the pandemic and treatment was still being established. But there are thoughts that some of these things, these thrombolytic complications, were quite prevalent, and they range from VTEs to myocardial infarction, stroke, and limb-threatening thrombosis. Importantly, the other thing is that we were seeing this in hospitalized participants, but we were also seeing multiple reports of late or post-discharge events that emerged. And these were in young people that seemed to be completely healthy otherwise than having the COVID. So this is the type of people that, maybe relatively young, had COVID, never really hospitalized, and then sometime, a few weeks to months later, had some sort of thrombolytic event. We were seeing the same thing about, hearing the same thing about people who were hospitalized and discharged and then seemed to be fine, no symptoms, but somewhat later having some sort of event as well. So these sorts of concerns led the NIH to consider what we wanted to do next. So, these profiles span both arterial and venous thrombosis, as well as micro and vascular. The rates that we were seeing ranged over a wide range of reports from 9 to 24%. This was primarily from observational cohorts, some starting in China and then others moving to Italy, at least when we were starting this. Those were the primary reports coming out. And given this hypercoagulable and prothrombolytic nature of the disease, the NIH and specifically NHLBI thought it was imperative to design both therapies and prophylactic strategies for these types of events. So that is when they approached us about working on these studies. Just also like to report that it is April 1st, so this is not an April Fool's Day, and it's springtime and it's snowing in Pittsburgh. So I just happened to glance out the window. So that's springtime in Pittsburgh. So let me jump into what ACTIV-4 is. It's Accelerating COVID-19 Therapeutic Interventions and Vaccines. So that's active, and there are, I think at this stage, 5 or 6 different actives. We are focusing specifically on ACTIV-4, which focuses on antithrombotic therapies. And what the idea behind ACTIV-4 is that 3 clinical trials were designed to evaluate the safety and effectiveness of various blood thinners to prevent and treat COVID-19 associated coagulopathy in 3 distinct populations of adults with COVID-19. And then we refer to these as inpatient, outpatient, and post-discharge or convalescent. So the basic idea here is that we're going to take people that are diagnosed with COVID and do clinical trials in 3 populations to either treat or prevent clotting events. So for an inpatient, these are people that are in the hospital and we're treating them on those preventing or worsening of clotting. Outpatient, these are people who are sick with COVID, but it was not felt that they needed to go to the hospital at that stage. And we're trying to prevent bad things from happening. And then the post-discharge group is those people that got sick with COVID, went into the hospital and were discharged. And now we're trying to prevent some of those long-term events that I mentioned earlier. So here's a little more detail on each of those. So for ACTIV-4, we have, like I said, what we call the inpatient, which is also known as ACTIV-4A. And ACTIV-4A is the population of COVID-19 positive hospitalized adults we stratify the population into two groups, the moderate and the severe. The severe group is those receiving organ support at the time of random assignment, and the moderate group are those that are hospitalized but not yet receiving organ support at the time of random assignment. Two treatments were being examined: prophylactic dose of heparin, which is usual care, and then there was a therapeutic dose, of a higher dose of heparin that was being evaluated. The outcome is 21-day organ support-free days. So what we do is look at the people that have been hospitalized and count the number of days that they are not receiving organ support during that 21 days post-randomization. I'll show you a little bit about the results later, but the basic idea here is it's an ordinal scale going from -1 to 21. The -1s, and this is just important to remember when we see some of the results, the -1 represent those that die during those 21 days. So it's an ordinal scale going from -1 to 21. Outpatient, oops, went too fast there. Outpatient, or otherwise known as active 4B, is the COVID positive adults not requiring hospitalization. So they've been tested, they test positive, and we're trying to prevent bad things from happening, right? So the population, the treatments here are there are 4 arms. There's a placebo arm, there's an aspirin at 81 milligrams, your basic baby aspirin. There's a apixaban at 2.5 milligrams, and there's a apixaban at 5 milligrams. The primary outcome is a composite outcome of symptomatic deep venous thrombosis, pulmonary embolism, arterial thromboembolism, myocardial infarction, ischemic stroke, need for hospitalization for cardiovascular or pulmonary events, and all-cause mortality at 45 days. So it's quite a large composite outcome. Next, we have what we call our post-discharge or convalescence study, and this is active 4C. And the population are COVID positive adults who are being discharged from the hospital. Treatment arms, only 2 in this instance, a placebo or apixaban. And the outcome is a composite outcome. It's the first occurrence of any of the components of the composite endpoint of systematic deep venous thrombosis, pulmonary embolism, or other venous thromboembolisms, ischemic stroke, very similar to the 4B. I'm not going to read the whole thing again. So it's the same, essentially the same outcome, except the timing is a little different. This is 30 days post-discharge. So those are the 3 studies that we have ongoing. And what I want to talk to you now about is, as we were preparing these studies, I want to talk about some of the novel clinical trial design considerations that we had to think about in putting these together. And I'm going to go through a lot of these, and the one thing that I'm not going to mention that is essential in the context of a pandemic was speed. We had to do this all incredibly fast, bringing a lot of people together very quickly to try to get these things up and running as quickly as possible. But the first one I want to talk about are just some of the study design issues that we took into consideration as we were doing this. The first thing we did was put together these in the context of a master protocol. And in terms of that master protocol, we had an overall design specified in the protocol. And then what we were able to do was separate appendix for each treatment arm. So for example, in the inpatient study, we had a master protocol that described how people would be randomly assigned, what the follow-up periods were, what concomitant treatments were, what the data collection system was, all that sort of stuff. And then each treatment that we were going to start was going to be an appendix. So the first study was to look at, as I mentioned earlier, the high dose heparin. That was an appendix to the overall protocol. Our original goal was to, as we were enrolling, add additional arms as the team evaluated potential other treatments. You'll see in a little bit where we stand, and we actually were able to end that first one early and start a new study immediately after. So the 2 protocols never overlap, but this master protocol does allow those protocols to overlap. The other thing is this second point is the idea of an adaptive design, where we're adding arms, like I said, for 4A. But the other thing is for 4B, that is the outpatient study, those that are sick and being followed, we have 4 arms. And based on our interim analyses, we can be dropping arms along the way. The other thing we're doing, and this is becoming more and more popular, is using, and I'm going to talk about this a little bit more later, especially with respect to interim analysis, is taking advantage of some of the Bayesian analyses to define decision rules for efficacy, superiority, and futility. So I want to talk about a multi-platform approach. Now, we talked about platform trials a second ago, but one of the novel things we did for 4A, the inpatient study, was a multi-platform study. And in the context of the pandemic, we had a bunch of people going out there and saying, this clotting in the hospital, this is an issue, we need to think about it. Oh, we should be looking at heparin versus normal prophylactic heparin versus high dose. How are we going to do this? And it turns out that a number of different agencies started this. So in the context of this particular study, we had 3 groups starting pretty much the same study. We had our group at Active 4 that was looking at this through the NIH funding. We had the Remap CAP group, which is a large multinational group that was actually developed around studying treatments of pneumonia, that very quickly pivoted to study COVID-19 and has been cranking out multiple papers on different treatment options. And then we had the Canadian Institutes of Health Research funding the ATTACK group to do almost the same thing. So it seems silly for all 3 of us to be working independently to try to answer the same question. So what did we do? We identified a mechanism to share information. So that was a lot of work. I would say a lot of work. I did some, but my colleague Mackie Neal did the vast majority of it. And I would say that what we had to do was, in summary, protocol harmonization. What are the treatments? What are the doses? What are the outcomes? And how are we all defining that? We were going to pool the data for analysis. So we had to do data harmonization. How are we coding the variables? What are the algorithms for the outcomes? How are we handling missing data? All those sorts of things. There was a centralized analysis. So coming to agreement on what that analysis plan was. We all came to the agreement of the same outcomes, but we had to do some slight modifications. And then we had to come up with a way to share resources. Who's going to be doing the analyses? How are we going to get the data to that group that is going to be responsible for the analyses? How are we going to manage all of that sort of stuff? Then there was I would say one of the more challenging things was the integration of the monitoring. So each of these entities had their own data and safety monitoring board. Because of this, we were reporting to, for example, the Active 4 DSMB, Active 4 only results in conjunction with the pooled multi-platform results. It created quite the complex reporting because the other DSMBs had to do the same approach. But what became even more challenging in that process was the idea of the DSMB's decision-making process. If we were to hit a flag for futility or efficacy, how are we going to work out this process if one DSMB feels that a threshold has been met and needs to stop, but another DSMB thinks we should continue? So there was quite a bit of time spent on working out that process and procedure and making sure that the DSMBs have the appropriate communication paths to do this. And then, as you might expect, there was quite a lot of discussion about the integration of reporting, the publication, the oversight, etc. So that may sound complicated. I think this figure will actually highlight that complication. So I won't spend a lot of time on this because it gets very complicated very fast. But basically, you have the multi-platform RCT right here. You have Active 4 feeding into it. You have Remap CAP feeding into it as well. You have ATTACK feeding into it. It's is a very complicated organizational endeavor. And I'm not going to, I could spend The first time we had this slide up for an hour meeting with the DSMB, it was a 5-minute presentation that took the entire hour, as you might expect. So I just wanted to put it up there to highlight for the group as to the complexity of doing these multi-platform. But it's an important thing to do in the context of the pandemic. The other thing I wanted to bring up was some novel recruitment pathways. So, traditional studies from NIH will have a coordinating center that contracts with sites, and the sites are responsible for enrolling participants. We've all probably done that and been involved with doing that for years and years and years. This can be a labor-intensive process of identifying the sites, setting up contracts, et cetera, et cetera. So when the pandemic broke and the NIH wanted to start Active 4, they came up with the idea of the network of networks. The basic concept here is the NIH has numerous existing networks. So why reinvent the wheel? That the coordinating center for Active 4 would not be recruiting sites, but we would be recruiting networks to participate in this study. And those networks would then leverage their existing infrastructure to speed up the process. Good idea. It's an interesting idea, and it could work. It leverages an advantage to this. It can potentially speed up the entire startup process. The con is that when it was initially looked at, the NIH said, well, we have 10 networks and each network has 10 sites, therefore we have 100 sites available. But the problem is there's considerable overlap among institutions. Johns Hopkins is in networks A, B, C, D, and E. So they're counted 5 times in that 100. So it doesn't really work. So what happened is, while we thought we had a large number of sites through this network of networks, we really had a substantially lower number of sites. So what we had to do is work to recruit additional recruitment pathways. And we look to enroll from a non-traditional point of testing facility. So the NIH was very hands-on in this process, and they're working to develop a relationship and a partnership with CVS to enroll individuals tested at their sites. It's a very different approach. And once again, we could have a group here come to talk to this group, spending at least an hour on the intricacies of trying to pull this off. But the basic idea is that people would go into CVS to get tested, and those that test positive are handed off at pharmacists at the University of Illinois, Chicago at the call center, and they describe the study there. And then if they consent, we make plans for a home blood sample collection from one of the local commercial labs to come get them. It's a very unique idea. It presents logistical challenges of really trying to consent people without talking directly to a physician at the point of testing positive. They do that through the call center. It also presented logistical challenges in the distribution of medications, because different states have different laws about how you can consent people for study and distribute drugs. So there were a lot of things to consider there, as well as the logistics of getting people out to take blood samples. The logistics come from 2 factors primarily. One being who actually is able to do this in the context of a pandemic, going to test people that are positive. And the other being that bandwidth. There's so much stuff going on right now. Did we have enough people to actually go out and do the collection? So it was quite a labor-intensive effort. The other thing that we wanted to point out in some of these novel experiences is low touch, and not only at the CVS, but even at the traditional clinical sites, there was low touch enrollment using a lot of pre-recorded presentations and electronic consent. Medications are given out at the organization where the person enrolls. But actually what happens is they are mailed from a central pharmacy directly to the participant, and they get enough drug for the entire treatment period. There's also innovative follow-up using RedCap/chatbots and the call center. Treatment initiation and adherence to medication is monitored this way. Symptoms and suspected clinical events are collected to monitor efficacy and safety. And then when clinical events are suspected, the pharmacist calls back to follow up, and then hospital records are obtained for adjudication by a central committee. Checking time here for a second. Okay, I got to pick up the pace. Some other things we have in terms of some novel approaches are understanding the biologic mechanism of the illness. So one of the things we did from the beginning is try to address this. And what we did is, in the context of a novel illness like COVID-19, the research community does not really understand the biologic mechanism of the complications associated with the illness. Really didn't understand who was clotting, who wasn't clotting, and why. So we went to basically so we understood the biologic mechanisms of the illness. And then if we did find effective treatments, understand the biologic mechanism of those effective treatments. So Active4 was prospectively designed to evaluate the understanding of these mechanisms, not only of the illness, but of the treatments. So each of the 3 protocols was designed to collect biological samples at least one point in time. Samples are sent to the Active4 biorepository at the University of Vermont. And then a separate funding mechanism that we coordinated through the Active4 Mechanistic Studies Center at the University of Michigan funded research projects using these samples. So, and I would just say that, take a step back, the repository at the University of Vermont is run by Russ Tracy, and the Mechanistic Studies Center at the University of Michigan is run by Jim Morrissey. Jim and Russ and I were all PIs of a trauma-induced coagulopathy study funded by the blood division of NHLBI, and that's how we got to be involved in all of Active4 at the very beginning. And just to give you a sense of what's going on, these are the first round of studies that have been funded through the biologic mechanisms. All very basic science, and a wide range of institutes participating from, you can see here, Michigan, Cleveland Clinic, UCSF, NYU, Colorado, MIT. Okay, moving on. One of the next interesting things we did was some Bayesian interim analyses. So one of the things for interim analysis for efficacy and futility are, we are very common in clinical trials. We are traditionally in most designs. There are many established approaches for these interim analysis. The most followed frequentist approach and therefore subject to multiplicity issues. So when we did this, we knew we were going to be wanting to look frequently. Matter of fact, we set up the process. Well, so what we decided to do is use the Bayesian approach, so we didn't have to suffer from the multiplicity issues. Therefore, we did multiple and frequent interim analysis without penalty, thus trying to get the answer as quickly as possible. So I'm going to give you the interim analysis approach for 4B. 4B is the outpatient study, just as an example. So at the interim analysis completion of the study, the posterior probability, the probability that the hypothesis is true, given the observed data and prior information, will be calculated. Each active treatment arm is compared to placebo using a multivariable adjusted logistic regression model. The a priori decision rules for superiority are that the posterior probability of the odds ratio of active versus control being less than 1 is greater than equal to 0.99. The decision rule for futility is that posterior probability of the odds ratio of active versus control being greater than 0.75 is greater than 0.95. So these were defined in advance. And we have a specific schedule to do these. And what we do is every month, and actually today is the first working day of the month, we lock the database and actually do an interim analysis for these studies. Now we do that for A and B, not C. C has a slightly different approach, but it is basically a monthly interim analysis approach. Let me give you a little bit about the current status of these. just to make sure that you understand where we currently stand. So for active 4A, we have some good news. Monthly interim analysis of the combined data from all patients have been performed, and we stopped randomization with a subgroup based on superiority or futility with respect to the primary endpoint. And these are the thresholds that we define for 4A. Superiority, 99 greater than 99% posterior probability of an odds ratio, proportional odds ratio greater than 1. And for futility, greater than 95% posterior probability of proportional odds less than 1.2. So what we found in both study, the interim analysis was done overall and stratified by severity. If you remember, we defined severe as needing organ support, moderate as hospitalized but not needing organ support. And that was specified a priori in the multi-platform interim analysis plan. So for the severe study, the severe subgroup, therapeutic anticoagulation did not improve hospital survival or 21-day organ support free days compared to usual care. And that was stopped for futility in December of 2020. That paper is now in press at the New England Journal and should be coming out shortly. The moderate study, the therapeutic anticoagulation greater than 21-day organ support free days compared to usual care, was stopped for superiority in January of 2021. Those data are still being analyzed and we're working on the paper. So I'm not going to show too much with that moderate group. I'm going to focus on reporting the results of the severe group, since those data are out and available. So remember that this was done in a multi-platform process. So what I'm showing here is just by treatment arm, the number that were enrolled in the severe cohort by platform. And you can see that REMAP-CAP was the big provider for this one. ATTACK only had 38 and active 4A had 133. I would just point out that in the moderate, it was flipped, and that REMAP-CAP provided a smaller group, whereas ATTACK and active provided a much larger group. Here's some baseline characteristics of the population. Average age in the two arms is about the same, around 60 to 61, about 70% male. Here you see the distribution by country. And this is somewhat reflected by the studies themselves. Active is primarily in the US. ATTACK, which was the Canadian group, was primarily in Canada. All this active and ATTACK both had sites in Brazil. Both came on board late, so they didn't really contribute much. REMAP-CAP was largely in the UK at the time of the data were frozen for these analyses. The other studies represented there are Italy and Spain, which came on very late and active. This is just to show you how severe the population was. You can see that the distribution of the organ support and the vasopressors is balanced between the two groups. But you can see that, baseline oxygen support was needed in pretty much the entire population. And the intensity obviously varied, but everybody needed that. And about 17 to 18% needed some vasopressors. Here are the outcomes. So by the treatment group, we had 529 people in the therapeutic arm that had a known outcome, and 543 in the usual care, 545 rather in the usual care. The median in terms of an interquartile range for the number of organ support free days was 3 and 5 respectively, going from -1 to 16. Remember that the -1 represents those people who died, okay? We treated it as an ordinal outcome, so the proportional odds ratio is 0.87, okay? And that 0.87 indicates that, those in the therapeutic anticoagulation group are doing slightly worse than those in the usual care group. You see here the posterior probability of 0.998. That is, we halted the study or stopped for futility because we have our a priori defined threshold of that posterior probability exceeding 0.99. So therefore, that's why in December we stopped this study for futility. And below you see the posterior probability for superiority. That's 10.6 rather. And if you recall, our posterior probability threshold for that was 0.99. So we're not even close to that. Just to give some perspective, we give the hospital survival discharge numbers below. You can see that survival was about 64% to 65%. Not a big difference between when the adjusted odds ratio was 0.88 for survival. Couple nice graphics to show you what's going on here. The graph on the left is a nice way of just showing organ support free days. So on the far left, you have what would be the dark red are the -1s, and that is death. Over to the far right, which I'm not good with colors, but I believe is blue. That's showing those that had 21 days of organ support free days. So you can see the distribution between the two treatment groups. They are pretty similar, not too much of a difference there. You can also see that if you take a look at the survival curve and you can see that the two lines, there's a little bit of separation right between 1 to 2 weeks, but not much. And they're pretty much on top of each other the whole way. Here are some other secondary outcomes that I just thought we'd share. You can see that there's major thrombotic events. The usual care group had a fair amount higher of major thrombotic events. The death rate was very similar between the two groups, and the combination of the two was pretty similar as well. The adjusted odds ratio was essentially 1. In terms of major bleeding, we had a pretty low rate of major bleeding in the two arms, 3.1% in the therapeutic group, only 2.4 in the pharmacologic group. I would like to point out that all of these major thrombotic and bleeding events were adjudicated by an adjudication center at Brigham and Women's Hospital. So all the data went there for them to adjudicate that they were truly events. Current status of the other studies that are going on, well, not even other, I'll start with Active 4A, which is the inpatient study. As you recall at the beginning, we said that we were trying to do this in adaptive design where we could add arms as needed. We were on the path of planning an appendix to our master protocol to study antiplatelet inhibitors. And the plan was to add those in as we were studying the anticoagulation treatments. For good news is we got answers to those anticoagulation answers before we could get this second arm added. So it's really being treated as a standalone study where we are studying P2Y12 antiplatelet inhibitors in the two groups, right, versus usual care. Like we did with the anticoagulation, we're stratifying by severe and moderate. And the difference here is we're comparing two P2Y12s to usual care, but that usual care differs between moderate and severe based on our findings from the anticoagulation studies. So we've modified what the standard therapy there is for anticoagulation and how you're going to use heparin. As of a couple of days ago, we had enrolled 116 people. Our goal is to get to around 2,000. Hoping we don't need 2,000 for the anticoagulation. We were able to answer the question before we got to 2,000. The other thing I would like to point out is that We are not doing this version as a multi-platform study as we did with the others. That for two reasons. One is the attack group that was created was funded by Canada, was designed specifically to test the anticoagulation treatments, and therefore isn't really set up to do P2Y12, but what we've done is rolled those sites into Active 4. The other thing is Remap Cap has a different, they have a different selection process for treatments that are going to be, and they're studying different antiplatelet inhibitors. And it's a slightly different protocol. So they really didn't line up like they did earlier. So it didn't make sense to do that multi-platform approach. For 4B, as of the 30th of March, we had 334 enrolled. And for Active 4C, as of the 30th, we had 9. And the reason for that big difference is Active 4C started much later than the other two. As a matter of fact, its first enrollment was in February. So it was by design, it's that low. It's nothing to be, well, it's always of concern, but it only just recently started. Some summary points I'd like to bring up. In the context of a pandemic, we have to take a variety of approaches to quickly provide answers and provide clinical guidance. Some of the approaches in this study are study management, while others are about design and implementation. In all instances, those need to be flexible. We need to be flexible in all of this. We're moving fast and we have to be as flexible as we can to try to get this done. Evaluations of these novel approaches need to occur quickly to determine if it works. You know, for example, in our 4B approach, we evaluated pretty quickly that the network of networks was not going to provide us a sufficient number of participants needed for the study. So that's why we went down that CBS approach. Also, I'd like to point out that there's a ton of people that work on this, a ton of people, and this slide does not do them all justice. There's a bunch of people at the University of Pittsburgh Coordinating Center. There are clinical leads for each of the 4 studies. 4A is led out of NYU by Judith Hochman and Jeff Berger. 4B is out of the Brigham and Women's with Paul Ritker and Jean Connor. And Duke leads 4C with Tom Ortel and Tracy Wong. As I mentioned earlier, the biorepository is out of the University of Vermont with Russ Tracy. University of Michigan is doing the mechanistic study. Jerry Krishnan is on the call, is leading the call center out of the University of Illinois, Chicago, and that's being used for 4B and 4C to do those novel follow-up assessments. And lastly, I want to point out that Scott Berry from Berry Consultants has been a key partner in helping us put together some of these Bayesian analyses. And with that, I will thank you, and I'm pretty much on schedule. So I think if it's okay, I will stop sharing my screen so I can try to see folks, and I can always pull stuff back up if needed.
Well, thank you, Steve. That was really very interesting and represents quite a feat getting all those people organized and working together. You know, as we know from other experiences, that what you all have done in under a year can take years to get done. So congratulations on that. I was wondering, I'll take the option to ask a question right away, is, so you presented results for severe and moderate, and I think they were both from 4A. So that was, you treated them basically as different trials by the stratum?
Yes, the a priori, the analytic plan called for the stratification, basically treating severe and moderate as separate entities and analyzing them that way. And that's just for the inpatient study, to be clear.
Okay. And I also wondered for the outpatient study, you know, where you're identifying people at CVS, do they have to be symptomatic at all or just test positive?
Test positive.
I'll give.
And usually if they're coming in, they are symptomatic or they just think they've been exposed. But yeah, all we really need is testing positive.
I'll give an opportunity for other people to ask questions or to type one in the chat.
Hi, Steve. This is Bob Wise. I'm curious how you develop the prior probabilities for the DSMB. Consensus of experts, data from other studies. How did that work?
It was a combination of the two, Bob. You know, there weren't a lot of other studies to really leverage. So it went primarily on expert opinion, but we tried to pull as much data as we could from the studies primarily that were coming out of China at that time.
I see. I have another question for you, and it stems from what's going on the ground, which is what I see. You're working up at the level of the systems and sites and institutions, but here at Hopkins, as in other institutions, we have a number of competing trials, all competing for the same patients. And sometimes there may be 8 people lined up at the door of someone in the intensive care unit, all trying to sign them up for clinical trials. And some of the trials don't permit people to enroll in other clinical trials, and it turns a little bit into the Wild West. We've made a number of institutional adjustments to kind of act as traffic cops, but I wonder if that was something that you faced, and if at the level of the coordinating center you had any input into that. And the second part of that is, what about this notion of somebody participating in several clinical trials?
So we are trying to, okay. So for the inpatient study, we are trying to treat this as very pragmatic. So we were permitting co-enrollment in other studies. The challenge we ran into there is a number of the other, in fact, other active studies, even those funded by NIH, were not permitting co-enrollment. So we had to do a little bit of work to try to secure co-enrollment. And it took a lot of approval processes within NIH and the respective DSMBs to allow that to occur. So we did do that. We also have run into, and I'm going to say this is more in the inpatient space than in the outpatient or convalescent space, where there doesn't seem to be as much competition. At least we haven't heard as much. We've been trying to work with institutions to just prioritize this study over others. It is really an institutional level decision. We try to support it as much as we can. And for one of the challenges there is for the 4B study, there is, I know of a competing study that is being done by pharma, and we've tried to provide reimbursement to the sites to be as high as we possibly could. We can never really compete with pharma and what they're paying, but we tried to be as competitive as possible. So those are a few of the steps we've taken.
Thank you.
Steve, there are a couple questions in the chat.
Yes. So did the meds need prior authorization? If yes, how handled by the central pharmacy? I may be referring to 4B CVS. So I'm not sure what you mean by prior authorizations, but they're both 4B and 4C are being conducted under an IND. So I think that helps. That might help some of the questions. The prior author, are you
providing the drug, I think?
Yes.
So you don't need a prior authorization from an insurance.
Correct. We are providing the drug.
Great, thank you.
Both 4C and 4B. And yeah.
And then there was a question about why only the apixaban at 2.5 milligrams was administered.
That was a long discussion because BMS is providing the apixaban, and so we had a long back and forth with them, and whether we do similar to what we're doing. And we actually initially considered doing the same 4 arms as for B, the aspirin and 2 doses of apixaban and placebo. But and this is where I wish my clinical colleague Frank was here, because he would answer this better. All I can tell you is that that's what the protocol design team came to. I was not part of those discussions.
Our colleague S. Renard has your hand raised on the Zoom. Would you like to ask your question?
Yeah, thank you. Steve, first of all, let me say before I ask the question that this is a heroic effort to get all of this done. And it obviously involved lots and lots of decisions. So one of them, of course, is the distinction between moderate and severe disease in the 4A study, which turns out to be really important since people in the moderate group who presumably are progressing towards severe disease do well, and the people in the severe disease do not, and if anything, trend poorly. So can you comment about whether that cut point, which was by necessity arbitrary, although it seems very sensible to me, whether that cut point in fact is at the right place to be treating the people who are going to benefit from the drug, and how that decision can be refined?
Yeah, that's a great point. So the decision to do that was very pragmatic. When we were designing 4A, it was thought we wanted it to be a very pragmatic study. So that's why we chose that. What we are doing now as a secondary analysis, I shouldn't say that now, it's one of our proposed secondary analyses once we get the analysis for the severe, the moderate cohort out, is to actually pool the data and actually look at these, looking at that definition in a more refined way to see if there are different thresholds that we should be using in defining where that cut occurs. So looking at specific organ support and maybe certain things matter more than just any organ support. So that is one of the planned secondary analyses.
Yeah. And what approach are you going to take to do that, since there's a lot of variables, and so multiplicity is going to rapidly get to be an issue in terms of trying to distinguish between trying to refine that decision point?
That's a great question. We have not got to that point in the process. Like I said, it's just been identified as a topic. We're still trying to get the other paper out. But I personally, I would look to maybe try to do some sort of almost a decision tree type analysis, because I think we might have enough of a sample that we can do some simulations to resample and maybe build a tree and test that tree to see where the appropriate thresholds would be. But that's just off the cuff, really. We really haven't planned it out in any detail as of yet.
Okay, great. Well, thanks a lot. Yeah, appreciate it.
Steve, I was also wondering on the CVS trial, when you said that you needed to go out and get blood specimens. So that must be, how's that limited geographically? What resource needs to be, who's going out and getting those blood specimens?
So we've contracted with Quorum Health, which is to go out and collect those specimens. And so it's limited geographically in terms of which cities we can do this and which areas, but it's also limited even within those cities because these the nurses are limited in the radius in which they can travel per company protocol. So yeah, it's providing to be a challenge for that and trying to manage that, but that was one of the things we're trying to work through. And we're working to expand that to bring going beyond Quorum to bring in other lab agencies like Quest as well.
And did you have to, for when you stopped those trials, did all 3 DSMBs have to agree on that? Did they meet together at all?
So the way it worked was it's active and remap cap the Venn diagram of the membership of those two DSMBs was huge. So so there's maybe one or two people that were on one but not the other, but the most of them were exactly the same people. So what we wound up doing is having those chairs attend the active 4 DSMB meetings to hear the discussion, and then we had to ask the 3 chairs to go off and discuss.
Yeah. Well, we're coming to the close. Is there any other questions for Steve? Well, I want to thank you both for this effort, which is amazing. And, you know, obviously a critical need for the world, and also for being willing to share your, to share it with us today. I think you showed a lot about how you can still conduct rigorous clinical trials that meet standards of evidence under very difficult conditions, and yet doing it quickly. I think it must meet the rule that, you know, what, and Bob knows it because he taught it to me. You can either do something that if it's fast and cheap, it's not going to be good. If it's fast and good, it's not going to be cheap. I imagine you might fall into that box.
I like that, and I agree with you. Yeah, it is not inexpensive, but I would also say it's just an enormous amount of people doing this. And just it's a wonderful group. Everybody is really stepping up.
Well,
So Steve, I just want to echo my thanks for speaking to us. It really was a wonderful talk today.
Thank you. Appreciate it. It was a pleasure to be here. I'm sorry I don't get to see you in person, but maybe soon we'll actually be able to do that.
I'll end the recording here.